Greetings to my lecturer. My name is Vidyashni Sivanesan, IS56185. The title of my research is Green Synthesis of Zinc Oxide Nanoparticles using High Viscous Subdiapal Leaf Extract and its band gap properties. My supervisor is Dr. Hanis Mohammad Yusuf along with co-supervisor Dr. Nurhana Bada. My research background. What are nanomaterials? Nanomaterials are particles with a very small size and dimension that is widely applied in industrial, health, food, and cosmetic fields. Among all the metal oxide nanoparticles, zinc oxide nanoparticles are one of the best utilized due to its large band gap, excellent thermal and chemical stability. In this research, green synthesis method is used to synthesize zinc oxide nanoparticles to reduce the hazardous impact towards environment. The phytochemicals in hibiscus sabdarifan leaf extract is responsible for the reduction of ionic bulk zinc oxide to formation of zinc oxide nanoparticles. The problem statement. Zinc oxide nanoparticles have been synthesized using physical and chemical synthesis routes such as sol gel, hydrothermal, spray pyrolysis, and precipitation. This method uses toxic reagents that are hazardous to environment, leads to imperfect surface structure of the nanoparticles, and also it is usually too expensive and requires advanced instrumentation. This research is significant because nanotechnology is an emerging field as it has a wide range of application, even it is also applied to human contact areas. So it is very important to avoid the use of toxic chemicals during the synthesis. Green synthesis of nanomaterials should be used extensively in the nanotechnology field. My research objectives are to synthesize zinc oxide nanoparticles via green synthesis by using high viscous subdiapal leaf extract, to characterize the obtained zinc oxide nanoparticles using PGA, FTIR, XRD, SEM, and UV-based analysis, and lastly, to examine the band gap properties of zinc oxide nanoparticles. Moving on to literature review, the researches conducted by Maditawa, Garpure, and Chan mentioned that zinc oxide nanoparticles can be synthesized in many variety of morphologies, which is due to the growth rate of certain directions. The common forms of zinc oxide nanoparticles are shown below, where A is nanobelts, B is nanocombs, C is nanoflowers, and D is nanorings. Besides, a study by Fakre also mentioned that green synthesis method produces nanoparticles with better defined particle sizes and morphology compared to physical-chemical method. This is proven by a research conducted by Arcas Chimal, where a Mexican plant was used in the green synthesis, and the results obtained showed the green synthesized nanoparticles is smaller compared to the commercial synthesized nanoparticles. The applications of zinc oxide nanoparticles includes in medical fields, cosmetic industries, and textile industries. In medical field, it is commonly used in drug delivery system, diagnosis and treatment of cancer, and bioimaging. The large surface of nanoparticles allows maximum contact between drugs and environment, and the small particle size is capable of penetrating the cell membranes and lead the deposition of drugs at the targeted site. Meanwhile, in cosmetic industries, it is often used in creams, ointments, sunscreen, and lotion. This is because zinc oxide nanoparticles are photostable and UVA and UVB reflector. This means the UV ray will be blocked from reaching the human skin. In textile industries, it acts as a good photocatalyst, which contributes to the photodegradation of polyethylene dyes. Talking about the hibiscus subdiapha plant, or more commonly known as roselle, belongs to the Malvaceae family. It is commonly found in the tropical areas in Southeast Asia countries such as India and Malaysia. According to a study conducted by Aduse, the calyces extract has highest number of phytochemical present compared to the leaf extract and stem extract, with phenols to be the most abundant and alkaloids being the least. Characterization of zinc oxide nanoparticles involves several techniques. Firstly, the PGA analysis is used to determine the thermal stability of the nanoparticles. FTIR is conducted to determine the binding properties and incorporation of biomolecule as well as the functional groups present in zinc oxide nanoparticles. X-ray diffraction is used to determine the crystallinity and phase distribution of the synthesized nanoparticles. SEM analysis is used to determine the particle size and surface morphology, and lastly, UV visible spectroscopy is used to determine the optical properties and conformations of zinc oxide nanoparticles. Talking about the band gap properties, zinc oxide has large band gap of 3.37 electron volt and high excitation binding energy of 60 mega electron volt. According to a study by Rusty, the nano dimension of zinc oxide nanoparticles shows an increase in pressure that leads to strong forces within the crystalline structure. Moving on to the methodology, the chemicals involved are hibiscus subdiapal leaves, ethanol, sodium hydroxide, and deionized water. Meanwhile, the apparatus are beaker, a glass rod, mortar and pestle, centrifuge tube, magnetic stirrer, Wattman No. 1 filter paper, heating plate, and the instruments involved are TGA analyzer, FTIR spectrophotometer, X-ray diffractometer, SEM microscope, UV visible spectrophotometer. The first step is the preparation of hibiscus subdiapha plant extract. The fresh leaf was washed with tap water to remove dirt and dust. Then it was washed again with deionized water and left to completely dry at room temperature for one night. The dried leaves were then grounded into fine powder. Then the powdered leaves were mixed with deionized water and was continuously stirred for 2 hours at 60 degrees Celsius. The extract was then cooled to room temperature and was filtered. The obtained extract was stored for the next synthesis. The next step is green synthesis of zinc oxide nanoparticles. The plant extract that was prepared earlier are being added to zinc nitrate hexahydrate while continuously stirring the solution for 1 hour. Then, sodium hydroxide was added to regulate the pH of the solution. A yellow color precipitate will be formed, and the precipitate was allowed to settle for 24 hours before centrifugation at 8000 rpm for 15 minutes. Then, the pellet was removed and filtered and dried in an oven for 2 hours at 80 degrees Celsius. Finally, a pale white powder will be obtained after drying. Then it is stored in airtight bottles for further experimental and characterization use. The characterization of zinc oxide nanoparticles are TGA analysis, FTIR analysis, which would be run at a wavelength range of 400 to 4000 cm, XRD analysis that would be operating voltage of 40 kV and a current of 40 milliampere, and copper K alpha radiation. Next is SEM analysis and UV-Vis analysis that would be recorded at a scan rate of 480 nanometer per minute by using quartz cuvettes. Lastly, the expected results of my research are the formation of zinc oxide nanoparticles in powdered form. PGA analysis should show a peak around 390 degrees to 400 degrees that indicates the formation of zinc oxide nanoparticles. The XRD analysis should produce sharp diffraction peaks at 2 theta values, indicating the crystallinity of the synthesized nanoparticles. The samples should exhibit UV-Vis absorption peak at around 370 nanometer that is attributed to the wide band gap of the nanoparticles. And lastly, the nanoparticles should form flower-shaped particles because the precursor used is zinc nitrate. These are my references that was included in my presentation. That's all from me. Thank you.